Communication between members of species are critical for both the survival of the individual and the species itself. Birdsongs have been studied due to their distinct differences between sexes. This has led to the discovery of the first neuroanatomical underpinnings of sex-specific behavior. Auditory differences in communication between sexes are common in the animal kingdom. However, most research has studied the attraction of mates or the defending of territories. Research needs to be done about the communication between offspring and parent. Offspring cry out when separated from their parents. This is common in many vertebrate species, including birds, rodents, and humans. It is not known if males and females vocalize this distress differently. FOXP2 is a transcription factor that regulates the expression or repression of many genes, including those related to neurite outgrowth, dendritic branching, and axonal morphology. The ratio of FOXP2 to other FOXP proteins can indicate whether genes are being expressed or repressed. The amino acid and DNA sequence and the distribution of FOXP2 around the brain are highly conserved in animals that communicate vocally. Mutations in the human FOXP2 gene can cause language and speech impairments. Studies have shown that FOXP2 levels increase during the period of song development in zebrafinch songbirds. Other studies show that chronic reduction of FOXP2 levels decrease the number of dendritic spines on striatal neurons. Birds with reduced FOXP2 levels cannot imitate songs well and have more variability in their songs. Data shows that FOXP2 affects the transcription of genes related to vocal communication in many animals. The purpose of our study is to determine if there are any differences in the ultrasonic distress calls of rat pups between sexes, and, if so, to determine what mediates that difference. It is also to determine if the dam perceives this sex difference and if it influences her behavior.
The subjects that we had in this study were pregnant Sprague Dolly rats identified on PN0, which was their day of birth, by injection of Indian ink onto their foot pads using a 30-gauge needle. They were housed in polycarbonate cages and underwent reverse light-dark cycles while being provided food and water. We also used fresh frozen human cortex samples. The demographic of the cortex samples that we used were from males and females. The males were all 5 Caucasian, and the females that we used were 1 Caucasian, 3 African American, and 1 Asian. The age range of these samples ranged from 3 to 5 years of age. All of the samples that we used were free of disease and infection and were taken from the left hemisphere of the Brodmann's Area 44. The methods we performed in this study were a Western blot analysis. Brain tissue from the PN2 and PN4 rat pups were blocked in 10% non-fat milk protein and then incubated in the anti-FOXP2 or anti-FOXP1 protein. The photo at the bottom of the screen gives you a representation of the Western blotting technique. We also used an ultrasound gate that was used for the recording of vocal emissions for a time-minute duration on the PN4 pups that were isolated from their mothers. Analysis of the acoustics was done by a SAS Lab Pro. A high-pass filter was used to eliminate background noise. Male and female rat pups were killed on PN4 and Western blots were performed on the cerebellum, amygdala, cortex, thalamus, hypothalamus, hippocampus, and preoptic area to detect the FOXP proteins. Each of the brain regions and samples were processed individually, and t-tests were performed to compare the males and females. As you can see in the results, FOXP1 was higher in females in the amygdala and preoptic area. FOXP1 was higher in males in the hippocampus, cortex, thalamus, and hypothalamus. The FOXP2 protein was found in higher concentration in males than in females in all regions except the hypothalamus and the preoptic area, where there were no statistically significant sex differences in the amount that was found there. In the pup retrieval portion of this experiment, in the control, males and females, males were retrieved first when scattered outside the nest, with a significance level less than 0.05. Interestingly, there was also a statistically significant difference in the control females and the females injected with the siRNA. The ones with the siRNA were retrieved much faster, since the sex differences were eliminated. There was not a significant difference between the control females and their siRNA male counterparts, or control males and their siRNA female counterparts. These results show that pup ultrasonic vocalizations can influence the order of retrievals from mothers.
FOXP2 is central to vocalization in many species, such as humans, birds, and mice. Our study showed higher levels of FOXP2 in males than females, but this was only found in areas of higher cognitive functioning and not reproductive areas. USVs are innate in rodents and indicate that the offspring needs the parents to come help them. Previous studies show that differences between low versus high rates of USVs caused by separation are affected by genetics. Our study showed differences between male and female USVs, though whether this is caused by genetics or hormones or the environment is unknown. Our study showed that males emit more USVs and that the USVs have a lower amplitude and frequency than female USVs. It also showed that siRNA-treated male USVs become more like irregular females and that siRNA-treated female USVs become more like irregular males. Previous studies show the preferential treatment of males from the mother. Specifically, one study showed that male offspring are groomed by the dam more than females, and that this was caused by an olfactory cue from the males. Our study showed that more males than females were retrieved by the mother, supporting that males receive preferential treatment from the dam. However, this was not caused by a male olfactory cue, as siRNA-treated females were retrieved more than siRNA-treated males, and as both anesthetized males and females were not retrieved by the dam. The USVs must have caused this difference in retrieval, showing their critical role in communication between the offspring and parent.
It has been discovered that there are gender differences in speech and language processing, but the origin and significance of these differences is unclear. It is commonly seen that female children are able to produce words before males and generally have a larger vocabulary. The common age that children start to develop language is around 4 years old. Because of that, we analyzed human brain tissue basal levels of FOXP2 protein from 4-year-old males and females. We also analyzed the expression of FOXP1, which is commonly associated with FOXP2. The FOXP1 exhibits delay in the onset of first words and often causes a smaller vocabulary of words, usually less than 100 by the age of 7. Sex differences have the potential to influence internal and external individual gender qualities. Here, I have provided a table to help organize the results of our study on the FOXP1 and FOXP2 protein analysis. For the rodents we tested, they showed no differences in the expression of the FOXP1 protein. However, for the FOXP2 protein, they showed higher levels of expression in males than in female rodents. For the humans that we used, they showed higher levels of expression in females than males for both the FOXP1 protein and for the FOXP2 protein. In this study of the zebra finch males, they had higher expressed levels of the FOXP1 protein shown than in the females. This result leads us to conclude that the FOXP1 protein is very similar to the FOXP2 protein and that it is associated with communicative sex. Our study also led us to conclude that male rat pups are more effective communicators using USV. The meaning of the differences in expression based on sex is not completely understood, and it won't be until we are able to get a better understanding of the precise role that the FOXP2 protein plays in humans and in rodents. Human sex differences in the FOXP1 protein and the FOXP2 protein and how they contribute to gender differences should play an important topic for future research to get a better understanding.
The study of sex-specific bias in maternal care can be translated to many other species. This could be useful in future studies to examine if the FOXP1 and 2 proteins impact this bias elsewhere. The largest application of this experiment was the relationship between the FOXP2 protein and communication. The FOXP2 protein only shows sex differences in areas of higher cognitive function in the brain, not in areas of the brain that regulate reproduction. This opens the door to examine communication differences in humans. FOXP2 could account for the origin of sex differences in communication. In 4-year-old humans, boys were found to have lower levels of FOXP2 than females. This is opposite than FOXP2 levels found in rodents. From this, it is hypothesized that higher levels of FOXP2 proteins are associated with higher levels of communication because this is found in both male rat pups and female human children. Examining FOXP2 levels in males and females may allow us to further understand communication differences in males and females to the point of improved communication between the sexes. FOXP1 was not treated with siRNA and did not impact sex-biased maternal care, so the measurement of it was not extremely relevant to the research. There were higher levels of FOXP1 in male zebra finches and female humans, showing that there is a relationship with FOXP1 and communication levels. We also know that there are increased levels of FOXP2 in male rats and female humans, and we know this correlates to increased vocalization, but we do not know why the genders for this aspect differ. In future studies, it would be beneficial to examine this concept in both the FOXP1 protein and the FOXP2 protein. Although it is harder to obtain and more regulated, more observation of the FOXP2 protein in human brains would be ideal to help support the studies of sex differences in communication.